For 4 decades, researchers struggled to decode the biological blueprints trapped inside Egyptian mummies. And for 4 decades, they completely failed. The blistering desert temperatures, the suffocating moisture locked inside ancient crypts, and the embalming chemicals utilized to protect corpses for eternity, all seemingly obliterated the one element capable of answering humanity's oldest mystery regarding Egypt's creators. Then, inside a pristine German facility, scientists isolated something that shouldn't have survived. Complete genetic profiles pulled from corpses entombed long before the colossal pyramids were finished. This unprecedented breakthrough instantly shattered a century of historical guesswork. The true architects of ancient Egypt defied every single expectation. Tracing the ancestral roots of ancient Egyptians was never an innocent pursuit. For over a century, academics fiercely argued. Some theorized the empire was seeded by Near Eastern invaders. Opposing voices maintained its origins were strictly African. This conflict became entangled with modern politics, colonial prejudices, and competing battles over historical ownership. Without DNA, the argument stayed permanently deadlocked. The first major attempt occurred in 1985. A young Swedish grad student named Svante Pääbo, working secretly at night in his university lab, tried extracting genetic material from mummified tissue. He announced a victory. The scientific community scoffed. Within years, his findings were dismissed as mere contamination. Contemporary DNA had polluted the ancient remains. The unforgiving Egyptian heat, it seemed, had permanently eradicated any genuine biological evidence countless centuries ago. By the dawn of the 2000s, the scientific consensus was absolute. Johannes Krause, a geneticist at Germany's Max Planck Institute, would retrospectively summarize it with brutal honesty. The vast majority of experts believed that pulling reliable DNA from mummies was essentially impossible. The extreme heat, the trapped humidity, and the harsh natron salts and tree saps utilized in embalming seemingly collaborated to destroy any preservation. DNA eagerly absorbs moisture. That water, mixed with intense heat and time, shatters molecular chains into unreadable, microscopic debris. For decades, this was undisputed scientific orthodoxy. Egypt's sands would forever guard their biological secrets. The breakthrough happened not in Egypt, but in modern technology. During the late 2000s, a revolution transformed the genetics field. High-capacity sequencing equipment, capable of reading millions of DNA shards simultaneously, phased out older methods restricted to targeting specific, isolated genes. Instead of blindly hunting for a needle in a haystack, researchers could now digitize a sample's entirety and sort it out afterward. More crucially, scientists finally learned to recognize the distinct physical fingerprint of authentic ancient DNA. When biological material degrades across millennia, it develops unique damage patterns. The chemical bonds capping every fragmented strand break in entirely predictable ways. Through a process dubbed deamination, cytosine bases naturally mutate into uracil. When sequencing machines scan these degrading molecules, they register as highly specific genetic typos, cytosine flipping to thymine, located exactly at the sequence edges. Modern contamination completely lacks this chemical signature. Only genetic material slowly rotting for thousands of years possesses it. This was the ultimate key, not merely extracting the code, but unequivocally proving its immense ancient origin. In 2017, Johannes Krause and an international team shifted their focus toward an archive gathering dust inside German museums for over a century. These mummies originated from Abusir el-Meleq, a Nile burial ground approximately 100 kilometers south of Cairo. Dedicated entirely to Osiris, god of the dead, the settlement served as a highly popular cemetery for centuries. Excavated in the early 1900s, the remains were shipped to the University of Tübingen and Berlin's Museum of Prehistory. The collection held 151 mummified skulls of everyday citizens, no pharaohs, no aristocrats. These were middle-class people, entombed within painted wooden caskets spanning 1,300 years of history, from the peak of the New Kingdom straight through the Roman occupation. Krause's team radically changed tactics. Instead of sampling the preserved soft tissue, skin, muscle, and organs that earlier researchers obsessively targeted, they drilled deeper into the dense bones and teeth. These highly calcified structures blocked out humidity, shielding the DNA from extreme molecular degradation. The results completely stunned them. From 90 of those mummies, the team recovered complete mitochondrial genomes. From 3 specific individuals, they extracted genome-wide nuclear DNA, the comprehensive biological blueprint inherited from both parents. Radiocarbon dating placed the oldest sample at 1388 BCE. The youngest dated to 426 CE. Between them lay the violent conquests of Assyria, Nubia, Persia, Greece, and Rome. Yet the genetic profiles barely changed. Across 13 centuries of foreign rule, the Abusir el-Meleq population remained remarkably stable. The DNA told a powerful story of continuity, not replacement. But the genuinely unexpected finding wasn't about stability. It was about origins. When Krauss's team compared the ancient genomes to global populations, both past and modern, a striking pattern emerged. Their closest genetic matches weren't in sub-Saharan Africa. They were in the Near East. The ancient inhabitants of Abusir el-Meleq clustered tightly with Neolithic and Bronze Age societies from the Levant. Modern Israel, Jordan, Lebanon, and Syria. They displayed a strong genetic affinity with ancient Anatolians from modern Turkey. Their Y chromosome haplogroups contain markers highly common in the Middle East. The implications rippled through the field. Modern Egyptians, by contrast, carry significantly more sub-Saharan African heritage, roughly 8 to 15% more than their ancient predecessors. This influx, the data suggested, occurred within the last 1,500 to 2,000 years, long after the Pharaonic era ended. The ancient Egyptians, it appeared, were genetically closer to archaic Eastern Mediterranean cultures than to populations deeper inside Africa. The 2017 study had limitations. All samples came from one Middle Egyptian site. The oldest dated only to the New Kingdom, millennia after the pyramids were built and the state unified. Critics argued the findings might not represent earlier populations or those from Upper Egypt near Nubia. 8 years later, that massive gap began closing. In July 2025, a team from the Francis Crick Institute and Liverpool John Moores University achieved the unprecedented. They sequenced the complete genome of an individual living during the Old Kingdom, the era when Giza's Great Pyramids were constructed. The man was buried at Newerat, a village 265 kilometers south of Cairo. Discovered in 1902, his body was interred inside a sealed ceramic pot within a rock-cut tomb. The pot and hillside created a microclimate stable enough to preserve his DNA for nearly 5,000 years. He wasn't mummified. Artificial preservation hadn't become standard practice yet. Ironically, that lack of chemical intervention may have helped. Utilizing shotgun sequencing on DNA extracted from the cementum of his teeth, researchers reconstructed a genome at double coverage, providing enough resolution to map ancestry with total confidence. The results mirrored and dramatically expanded earlier findings. Roughly 80% of the Newerat man's ancestry traced back to archaic North African populations. The remaining 20% linked to something totally unforeseen: genomes from the Eastern Fertile Crescent out of Mesopotamia, modern Iraq, not the Levant. The genetic modeling was sharply precise. The best statistical model revealed 77.6% ancestry matching Middle Neolithic populations from Morocco, themselves a blend of Levantine farmers and older North African groups. The leftover 22.4% aligned flawlessly with Neolithic Mesopotamian genomes dating to 9,000 and 8,000 BCE. This connection wasn't a secondary mathematical fluke. Statistical tests proved direct biological affinity with Mesopotamia itself. This suggests migration flowed from the Eastern Fertile Crescent into Egypt during or before the early dynastic period. The routes remain fiercely debated, possibly along Mediterranean sea lanes, through the Red Sea corridor, or via unmapped intermediary populations in the Levant. This genetic proof finally confirmed what archaeologists long suspected from cultural artifacts. Pottery styles, cylinder seals, and architectural motifs echoing Mesopotamian originals were appearing in pre-dynastic Egypt. This exchange wasn't merely moving objects and ideas, it was moving human beings. The Newerat man lived during a pivotal era. Radiocarbon dating placed him between 2585 BCE and 2570 BCE, perfectly overlapping the transition from the early dynastic period into the Old Kingdom. The first pyramids were rising. Upper and Lower Egypt had recently unified. The bedrock of history's longest-lasting civilization was being laid. His skeleton narrated its own deeply personal story. Bioarchaeologist Joel Irish calculated his death between 44 and 64 years of age, extraordinarily old for that brutal period, equivalent to reaching your 80s today. His bones showed extensive osteoarthritis and muscle development, consistent with decades of hard physical labor. His height was a modest 157 to 160 centimeters. Genetic readouts predicted brown eyes, brown hair, and dark skin, common features among present-day Egyptians and North Africans. He was born in Egypt. Isotope signatures inside his teeth flawlessly matched the Nile Valley's diet and climate. This was no foreign merchant or diplomat buried far from home. He was an Egyptian. His genes, however, told an epic story reaching far beyond the Nile. Naturally, these breakthroughs do not settle every question. They sharpen some and open others. The Newerat genome represents one solitary individual from one site at one specific period. Egypt was unimaginably vast. Upper Egypt, positioned closer to Nubia, might reveal different genetic patterns. The northern delta region, exposed to Mediterranean trade, likely absorbed different demographic influences. Constructing a complete genetic history of Egypt requires dozens or hundreds of sequenced genomes spanning millennia across the entire length of the Nile. Some scholars have questioned whether the Abusir el-Meleq site utilized in the 2017 study drew populations with highly unusual ancestry, possibly descendants of foreign mercenaries or settlers granted land in the Fayum region during the New Kingdom. Others argue the sampling remains too limited to draw sweeping conclusions about all ancient Egyptians, particularly those from the earliest pre-dynastic periods when the culture's foundations were being laid. The fierce debate continues exactly as it should. However, what the DNA has established beyond reasonable doubt is this: the ordinary people who built and maintained Egyptian civilization across its first 3,000 years were not replaced by waves of conquerors. Their genetic profile held remarkably stable despite the reigns of foreign dynasties. Persians ruled, Greeks ruled, Romans ruled, yet the native population endured unchanged. The foundational ancestry of these ancient Egyptians was a complex blend, predominantly North African, with deep roots in populations that occupied the Nile Valley and the Sahara when the desert was green, but also carrying a significant component from West Asia, specifically Mesopotamia. This mixing likely occurred before or during the earliest phases of Egyptian state formation. It wasn't a later import, it was present at the absolute foundation. Modern Egyptians, the data confirms, retain approximately 80 to 90% of this ancient genetic signature. The changes that occurred, the additional sub-Saharan African ancestry, came later within the last 2 millennia. The unbroken continuity is vastly more striking than the change. 40 years ago, Svante Pääbo stared at fragments of mummy tissue and dreamed of decoding the genetic story of ancient Egypt. His early results were quickly dismissed. The blistering climate, the caustic chemistry, and the sheer passage of time seemingly erased the record forever. Today, Pääbo holds a Nobel Prize for sequencing the Neanderthal genome, and the mummies he once studied have finally yielded their secrets to the next generation of technology he helped inspire. Inside a sterile German laboratory, resting in a Liverpool museum, and buried within the tooth of a man who labored beneath the Egyptian sun nearly 5,000 years ago, the code miraculously survives. It tells a saga vastly more complex than any single narrative about modern race or origin. A cinematic epic of populations moving, mixing, building, and then enduring across millennia while empires rose and fell around them. The ultimate secret locked inside those tombs was never about who the Egyptians were. It was about how long they remained. What are your thoughts? Leave a comment below. Make sure to subscribe for more.